A lot of familiar faces. I'm happy to get together with this group, and I think it's going to be a pretty fun meeting over the next few days. And I usually when I travel around the world, I always have some frightening slide about our ice and snow and huge snowdrifts and everything else. So everybody's going to feel I'm like a chronic liar now. I would be remiss if I didn't thank Annie Savoie from the livestock feed section really for being the genesis of this at the task force on novel foods and feeds, when she sort of planted this idea with ILSI colleagues, and we've been working on this, as Cameron alluded to, we've been working on this topic in Canada from our own perspective. So it's good that we've got a little bit of international uptake on there. We're also working with U.S. colleagues, and the more involvement we have and the more science we can put in here, the better. And I realized that despite your comment, I got 40 minutes here to talk about unintended effects, and I thought, what am I going to talk about for 40 minutes? So I thought I'm going to give you a little bit of a risk assessment context. Now I'm looking around the room, and I see Ellen Raybould sitting at the back here, and Alan Gray, and Andrew Roberts, and the rest of the people in here, and realize, well, God. So you can amuse yourself today by counting the number of times I make mistakes and figuring out where you're going to correct me later, and all the misstatements and all the things I may have missed or left out. So I'm going to start talking about where we really came from with the regulation of biotechnology, because we started off with kind of a scientific perception, and I think we may have really drifted a little bit from there. How Canada's take on the regulations, where we came from, definitions, because I was asked to provide definitions, and they're always fun for people. And I'm going to go through some risk assessment concepts to maybe set the stage here as we talk about unintended effects, how we address unintended effects, a little bit about why we're here, what really the genesis of this was, and then some conclusions. Okay. So where I think a lot of us started off with regulatory process, and I'm involved with the OECD, and Annie and others, Luke in the room here. We have an involvement in the OECD, and the OECD was really one of the first places where a lot of these concepts about how to look at risk assessment really came from. So the blue book, and if Sally McCammon was here, she'd be, it's almost like the Bible for some of the people who were around the early days of the OECD. And it was really an output about recommendations on DNA safety that came from scientists, academia, some of the advisory committees, regulators, government, and industry. And then the next iteration of this was really science-based regulation for GMOs, the next steps. And it outlined a lot of the concepts that we've sort of taken on board today when we start talking about how we do risk assessments of these products. So it outlined some of the safety assessment guidance for products of biotech, and it captured some key principles, such as basing the safety assessment on comparison with the unmodified counterpart, and the focus on the new trait in the plant. It did, however, avoid defining what biotech was, an area which, really in Canada we have one kind of definition of biotech which is a little different, and I'm going to talk about that. And mostly this focused on the environment, but it really was meant to be broader based kind of concepts. So where Canada went to was in 1993, we articulated some of the key principles for, this is a biotech regulatory framework. And embedded in here is really guidance for the risk assessors. And it really talks about how we think about the technology, how we mean to regulate it, and what we think about. The idea that we use existing legislation, that these products are looked at by the people who would normally look at them, whether they're feeds or foods or plants or seeds in the environment. The same legislations apply, and the same people are doing the safety assessments. So a clear articulation that the regulation should be based on the product, not the process. And the framework also recognized that regulations should be put in place that allow innovation to proceed while protecting the health and safety. So that tells us where that regulatory bar is set. It's set at in such a place where we're still going to allow, we're going to realize that we're going to have to take on a certain level of risk. No activity is risk-free. And that's really a pretty clear articulation of that policy. So Canada went one step further. And when we talked about how we think about biotech, we really talked about biotech specifically, that they're not specifically plants modified by recombinant DNA. And the advice that came back from the science community was, really, it doesn't matter how you get there, it's the product. It's not genetic engineering that's risky, it's the kinds of things that you can produce, whether it's genetic engineering or conventional breeding or whatever. And the example I always use is herbicide-tolerant plants. So when you go out in the world and people talk about, triple herbicide-tolerant canola superweeds, the superweed, one of those herbicide tolerances is really only regulated in Canada, and that's the Clearfield ones. The rest of them, they escape regulation in the rest of the world. So we have in fact varieties of canola that have been produced, herbicide-tolerant canola, broad-spectrum herbicide tolerance, produced by all those methods. So I got some definitions here, and what better place to talk about risks than the Canadian Centre for Occupational Health and Safety. So anytime we talk about definitions, it's always a thorny area, but these are some definitions that look kind of convenient. So hazard, any source of potential damage or harm. Risk is the probability that the harm is going to result, and harm is the negative outcome for the hazards. That's just to be really clear, because people tend to use a lot of these terms interchangeably. And this is the classic expression of risk assessment is risk equals hazard times exposure. I know that the EPA has a little bit different, and their probability outcome, and the OGTR talks about this in different terms. But these are kind of some global expressions, and they're iterations really of basically the same kind of concepts. Our starting point for when we think about how we're going to regulate these things is our conceptual model. And again, I went to a definition right online. A descriptive model of a system based on qualitative assumptions about its elements, their interrelationships, and system boundaries. So that really talks about how you're going to think about that. You think about your new plant, how you're going to think about your new plant in whatever context it happens to be. But it really it gives you boundaries for that, and it gives you something to model inside of. It gives you some sort of scope and box to put things in when you start thinking about it. Our conceptual model for biotech crops is developed over many iterations and forms the basis for our assumptions during the risk assessment. And for example, for an environmental risk assessment, my area of knowledge, the conceptual model considers the crop, the new trait, the likely receiving environment, and pathways by which the effect's going to occur. So those are pathways to harm. So what are your routes of exposure, things like that? Where is it going to go? What's the receiving environment? What is your new trait? And those are the same concepts that you really thought about in terms of the OECD. It's crop, trait, and receiving environment. Those are the considerations, and this is really an iteration or an articulation of the same kinds of concepts. Integral to the conceptual model is our assessments use a familiar counterpart as a reference. And I'm going to give you a definition of familiarity based on the OECD. And it relies on substantial equivalence to define acceptable harm. So when we think about those terms in terms of the environment, Canada's a little bit unique in using substantial equivalence, not for the food and feed side, but also talking about the environment. And substantial equivalence is usually something that'll set off a firestorm of discussion, controversy, and there's been some authors with different viewpoints. These concepts are adapted from OECD food safety assessment criteria and we apply, like I said, across the board. And they're very effective and have proven very effective for us for crops and traits that we know well. These are concepts that really do work, and we have a pretty good history in Canada of safely deploying these crops over really a relatively long period of time. Familiarity, it's the knowledge of the characteristics of a plant species and experience of use of that plant species in Canada. So we have a reference point. When we have a risk assessment, we're not going back and looking at canola all over again or maize all over again. We have reference points. We have knowledge of these things. The biology documents that we create in Canada are the basis for our comparative risk assessment. So we articulate, just like the OECD consensus documents, we articulate what the biology of that species is in Canada. And that gives us the basis for that comparative risk assessment. So when you're looking at effects, intended or not, that's your basis. That's your baseline for that assessment. And the knowledge of the agricultural ecology is the assessment context for the crop species. So we think about those things in terms of being a cultivated species. So substantial equivalence, the concept of comparing new products with existing familiar products. So when we start thinking about it, I always use this example, close your ears, Paul, but when the Royal Society came in and they did a review of biotechnology in Canada, our regulation of biotechnology, Spencer Barrett from the University of Toronto asked me, how do you look at the effects on biodiversity of Roundup Ready canola? And I said, well, how do you look at the effects of biodiversity on growing canola? We grow something like 8 million acres of canola every year in Canada. Anybody who flies across the prairies can see it's a sea of yellow. You can see the pollen scum. I mean, right? Alan is a regular visitor to Alberta. So that's our context. So when you think about that, when you add that Roundup Ready gene in, what does that really mean? So what is the difference in the effects on biodiversity, both on the scope of putting that gene in the context of the fact we already grow 8 million acres of canola and its substantial impact on biodiversity, but that gene inside the plant. Our canola plant has something like 30,000 genes in it. How does that fit into that whole scope and context of those genes and those interactions? Plants do all kinds of things. They're little chemical factories. Their interactions are both negative and positive. So when we think about it in that context, what does that Roundup Ready gene? How does it fit into there? So for us, that really gives us our scope. That's really our scope, that substantial equivalence. That's really where our scope is. In the agricultural context, it should be as safe as. It should be in that same kind of context of interactions. So our risk assessment considers the relative safety, that's our comparative safety assessment of the product in both the short and longer term. So again, the information about the novel plants compared to information about biology of the counterpart. And it considers the expected trait in the context of the unmodified plant and considers how it can affect things we value. So what about the things we care about, like monarch butterflies or bunnies or people or animals? And by using indicators we can measure. Plants disappearing, butterflies dropping out of the skies, bunnies dying. So this is really how we conduct our assessment. And in this structured analysis, though, how do we account for those unknown unknowns, which could include unintended effects? So we have a structured basis for looking at this. We have a good comparative risk assessment. We have the context of substantial equivalence. But how do we look at unintended things that we didn't intend? This is really focused on looking at the things that we intended to do. Genetic engineering is conducted with a specific objective in mind. The aim is to transfer potentially foreign DNA, could be the same DNA, into a host organism and affect specific changes within that organism. So you want to do something. You're doing it for a reason, generally, even if it's research. And for example, the plant may be meant to become herbicide or pesticide resistant. So a Roundup Ready crop or a BT corn. Those unintended effects are those that appear in addition to the intended results. So this is really where a lot of the conversation rises around genetic engineering. It's the what ifs. What if you have your altered version of the enzyme in a metabolic pathway has an effect on related pathways? What if you have some amino acids that are backing up? Or what if it ruins a feedback loop? Or what if the insertion of your new gene disrupts an endogenous gene? You get a read-through that creates a chimeric protein. So what if it pops in someplace and it's not properly terminated and it reads all the way through and it's got plant DNA and you have some chimeric protein? Or what about a downstream effect on another gene? You've got these strong promoters. What about something, it's affecting something downstream or something in a gene family, because a lot of these genes co-evolve. Or another gene or a cryptic open reading frame. So these are some of the discussions that have centered around unintended effects. And these are things that people have wondered about, but in real terms, we don't really see. They're always possible. As scientists, we can come up with all kinds of really good scenarios, but they're not things that we really see often in practice. So we know that unintended effects are possible. It's always possible to do things like that, but how does this fit into our risk assessment approaches? So many regulatory systems consider unintended effects important enough that they regulate every transformation event. So a lot of, for example, the Codex guidance is really about every transformation. Or if you look at the EU guidance, it's not just about every transformation, it's about every transformation. And it's also about stacked events, the things that can happen if you have unintended effects, and then you breed these things together and create even more unintended effects. So how do we address these types of phenomena in the risk assessment, and how important are they? Where do they really fit into the context of when we think about that our risk assessment, which is really focused on the things that are intended, how much focus do we need to put in the things that are unintended? So back to my risk assessment, and I pinched this slide off the internet because really, it doesn't say hazard times risk, but really always the risk assessor back there, probably Alan, turn around and say, well, the crocodile can always hop out of the pen, and then it's almost no risk. So, but really in real terms, it's the same idea. The risk is a function of not just having the hazard there, but also having exposure to the hazard. So the first step in the risk assessment generally is the hazard identification. And usually hazard identification is really a key part of how you're going to embark on your risk assessment. And really, at this point, a failure to engage in proper hazard identification can compromise your entire risk assessment afterwards. And this is where a lot of that speculative stuff comes from, where you can really imagine things that could happen, but really haven't really got a strong hypothesis about how that's going to happen, or what it's going to affect, or what you're really thinking about. And this is back to what I talked about, that you want to think about what is it you're trying to protect, and what possible harm is going to happen to it. So Endpoints are value-driven and set by policy. So, for example, the protection of endangered species. That would be a protection goal, but the process to assess harm to an endpoint should be firmly based in science and hypothesis driven. So to say, for example, in Mexico, we don't want to have any transgenes in maize, may be a valid protection goal or a valid endpoint, but really what the important part here is it possible to have that harm occur? And comparative safety assessment should be based on reasonable risk hypotheses. So really, when you think about that comparison, you really want to think, what is my potential route to harm, and is that really a possible occurrence? And just having a difference doesn't necessarily mean a harm. And that's one of the real evils of genomics. Because you can go back in there and you can find all kinds of things, and the transcriptome's different, or we can find little alterations around junction sequences, but that doesn't necessarily mean a harm. We need to have a route or a pathway to harm. There needs to be some sort of realistic risk hypothesis that connects to that. Difference doesn't mean harm. We again, back to our substantial equivalence, we really have a context for how we're going to think about those things. And really, in the whole scope of our comparative risk assessment, where do those things fit in? Where is the evidence of harm? Exposure assessment's a determination or estimation of the route, frequency, and duration of exposure. And again, whoops, there's my reference point. For unconfined release, for us, when we talk about really that's what I figure we're talking about here today, is unconfined or commercial or deregulation. It's exposure generally considered to be uncontrolled in the environment, so no longer subject to conditions. So it's now off, and you can put it into commingling, you can feed it to animals, you can use it as food, you can do whatever you want. And although some of our data is quantitative, for example, the toxicology data we use for looking at effects on targets or non-targets, the assessment's primarily qualitative. When we really think about that in real terms, our endpoint is it's as safe as conventional corn. So when the minister's office came to me and said, can you tell us that Roundup Ready alfalfa is absolutely safe? And I can say, well, it's as safe as conventional alfalfa. Because it is. Because, it's like all these things. It's contextualized. Growing alfalfa, well, it reduces biodiversity. It can have effects on other things. You're going to apply whatever your management techniques are. So really, these things are all in terms of your counterpart. All in terms of recognizing that agriculture poses a risk to the environment, and it's that context that we're working in. The comparative risk assessment proceeds in a structured manner based on your risk hypotheses. So you're going to go through there and look for reasonable pathways to harm, postulate your risk hypotheses, and then look and see if there are possible routes. For tiered approaches, laboratory tests will often identify the worst case scenario. I know, York, I know that's something you're a true proponent of. So if you think about this, if it doesn't kill something in the lab under controlled conditions with maximum exposure, it's not likely to kill something out in the field either. So you can really come up with a realistic proposal from here. So however you're managing to conduct your laboratory tests, it's usually a good route to avoid some of that next tier testing. So if the first tier of testing doesn't identify a risk, then the assessment can stop. And that can apply to all your risk scenarios. So if you're looking at this and you're not getting any hazard in the lab, then you don't need to go any further. You can stop right there, even if it makes you comfy to go off and do field trials. And this data can be quite portable. So if I don't kill caterpillars in a lab in Belgium, I'm not likely to kill caterpillars in a lab in Mexico or a lab in Canada either. So we can take advantage of that. Yet we tend to redo that data often over and over again, but we really can take advantage of that. Good laboratory practice is good laboratory practice. Good toxicology is good toxicology. If somebody's done the experiments properly and they're properly controlled, you can take advantage of that data. So for us, for unconfined release, and I've given the example of the environmental assessment here, and we call these our assessment focuses on the novel trait, the novel plant characterization, its interactions, and any changes to agricultural silvicultural practices as it affects our 5 pillars. And these are altered weediness, gene flow, plus potential impact on non-targets, and potential impact on biodiversity. So that kind of structures your whole risk assessment around the outside. But again, your assessment itself still focuses on those key criteria. Risk assessment itself concentrates on the intended trait, where unintended effects can be anticipated. So, for example, do you have a realistic expectation that there's going to be a change to the metabolic pathway? Or interactions with endogenous proteins in there? Is there some possibility is that a likely scenario? Risk hypotheses can be constructed. So you really have a way of getting at those kinds of things. Unexpected effects are more difficult to address. So this is the whole idea of gene disruption somewhere else, or something that you can't predict, or something that's really unpredictable. So generally what we have tended to do is lean on phenotypic characterizations. So one of the reasons that we spend a lot of time on agronomic data, and I think Stephen Yarrow, who's back there, coined some expression about looking like a duck and walking like a duck, but it's really the same thing. If it looks like a canola plant and grows like a canola plant, then we can figure it's a canola plant. And if there's any other disruption somewhere through there, well, we can't look at all 30,000 genes, all the metabolic pathways in there. We're not going to look at all 30,000 genes, so we're going to have to rely on some sort of an indirect measure. And that tends to be where we've gone. And in real terms, we don't really tend to see very much out of this. And genomics can lead risk assessors down unproductive routes. And often, you'll try and compare transcriptomes and look at different kinds of things. And one of the great things about genomics, they're fabulous research tools. They're wonderful ways of generating enormous amounts of information. But unfortunately, there are wonderful ways of generating enormous amounts of information. And a lot of that information, really, you can't really look at. It's really difficult to apply it to anything without a hypothesis, unless you've got a clear hypothesis about what it is you're actually going to do with it, what you're actually going to do. I can go out. So, for example, Jamie's former boss, Brian Mickey, did some really wonderful transcriptome analysis, looking at exactly this idea of unintended effects, to went out and look and see what happens to the transcriptome. Started with a clear hypothesis. Spent a lot of time validating a lot of his assumptions. Came up with some generally useful data. Generating transcriptomic data and finding differences doesn't lead you really anywhere. Weight of evidence is something that we bandy about a lot, at least in my world, that we always talk about how we use a weight of evidence approach, and we talk about weight of evidence. And the only place, when I went and actually looked for a definition of weight of evidence, the only place I found weight of evidence was in legal texts. Lawyers use weight of evidence a lot. We talk about it, but we don't really have a structured way of doing it. So based on our data and referring to risk hypothesis, we need to come up with reasonable conclusions. So, we can spend a lot of time. One of the things about risk assessment, it's not a research project, it's a risk assessment. So as it's not a research project, you are not going to have, ever, all of the answers that you want. So what you're going to have to do is come up with some reasonable conclusions based on the information and the data that you've got. This implies an examination of all your available evidence, positive and negative, a consideration of the quality, and weighing studies to arrive at a reasonable conclusion. And I don't think we, at least in Canada, have not really articulated what kind of standard of proof we require, which is why we spend a lot of time running around looking at unintended effects, because we're never really comfortable. We had a talk with Paul Keyes this morning, and we talked about some of the weed risk assessment models. So I have a group of weed risk assessors. So they can spend 2 weeks and do a risk assessment on releasing a new species with 40,000 new genes into the environment, and we can spend 2 years doing a risk assessment on a corn plant that's got 1 new gene in it. So, we really have to start thinking about what kind of standard of proof do we really require, and I don't think we really know. So how relevant is a piece of data to making a hypothesis probable? That's a consideration, reliability, and sufficiency. What is our threshold of weight? So what is actually necessary and sufficient inside a risk assessment? And that's one of the problems with the policy envelopes that most of us operate inside now, is we don't really have that kind of guidance. So risk assessors, especially if somebody comes new to there, you don't really have the same kind of perspective. We spend a lot of time, at least inside my group, mentoring new risk assessors, because people don't really know. We want to look at effects on biodiversity. Well, what does that mean? These are big things, and people are making big policy decisions through there. What kind of information do you need to come up with realistic decisions? It's important. Those are important discussions. And it's important that really, from a risk assessment standpoint, there's a bit of a policy vacuum around the outside of it. And that really comes back to our famous regulatory statement, is that need to know versus nice to know piece. These are things that, are you asking that question? And that's certainly the precision that we have inside our group, and I'm sure others as well. Are you asking that question because it's absolutely essential to inform that risk hypothesis, to come to a conclusion, to inform your final judgment, or is that something that just makes you happy to know? One of the problems is many of us come from research backgrounds, and as a context, we're curious. We're naturally curious. People in this world tend to be scientists. And as scientists and as curious people, you ask questions. And if you come back and you ever really challenge people, do you really need to know that? Why? How does that fit into your risk assessment? And in a lot of cases, risk assessors don't even know what kind of assessment endpoints they're looking at or what kind of endpoints they're trying to address. So in the context of our conceptual framework, it's difficult to predict but not impossible. So how we use weighted evidence. And as an iterative process to rely on experience and previous data to build a body of evidence that we can rely on. And I don't think we're really doing that. So we can take our experience, we can employ that into our conceptual model, we can use weighted evidence, and it gives us the option really of looking at a whole lot less data. And maybe being a whole lot less concerned about some of these unintended effects because we certainly have the experience dealing with them. So we asked for a wide variety of data for all of us. And I mean, the data packages, and I can see some developers in the room here, the data packages from my early days working at the agency have bloated considerably. I mean, it's simply because it's so easy to generate a lot of this data now. You know, we're looking at a lot of next generation sequencing data. We have a wide scope of information that we never even thought to ask for before, which is now bloating out a lot of our dossiers. I think I'm... Okay, so putting unintended effects into context, I'm mindful of time here. Putting unintended effects into context, there's a scientific consensus that genetic engineering is not risky. So I went through and I looked for some salient terms and something that, I mean, there's any number of different articulations by any number of different people that say fundamentally exactly the same thing. So this is from the American Association for the Advancement of Science Board report from 2012. I welcome you to go and look at it. And this is in response to some of the things, the conversations around GMO labeling or G-food labeling. So the World Health Organization, the American Medical Association, the U.S. National Academy of Sciences, the British Royal Society, and every other respected organization that has examined the evidence has come to the same conclusion. Consuming foods containing ingredients derived from GM crops is no riskier than consuming the same food containing ingredients from crop plants modified by conventional improvement techniques. I mean, it couldn't really be any clearer. So when you think, oops, holy crap, what have I done now? What have I done now, Nancy? Here we go.
There you
go. So, I mean, that really, there you go, that finished. That really couldn't be any clearer, and I really think that probably says it all. So this is where we started off. If you go back to where we think about, you know, the early days, the OECD, the Blue Book, 1992, that's really where everybody started. Thinking exactly that. If you think about our first round of scientific consultations occurring in the 90s, that's what our scientific community came back and said too. It's the product. You know, this isn't any riskier than what we're doing. This is really about the product. And it doesn't mean that, you know, you can't create bad things by any kind of technique. It's the product. So why have we spent all this time focusing in on one kind of product? And the answer is pretty simple. Because we can. So can we learn from our experience? Increase familiarity with plants, foods, and feeds. We have lots. We've been doing this for a long time now. I mean, the first authorizations in 1995, at least for us, and I think earlier in the U.S. You know, here we are in 2013. I mean, we've really done a lot. I mean, we've got, I think we're up to about 90 crops authorized for unconfined release now. So I mean, it's not like we're new at this, and we haven't really seen this pantheon of disasters. There are a lot of meals consumed by billions of people around the world. You know, a lot of people are in this business now. There's a growing body of scientific research on genetic engineering. There's a lot of information that's out there. We know more about genomics. We know more about the genome now than we ever did. There's been advancement in molecular analysis techniques. We have an unprecedented understanding of plant genomes and genetic change. You know, really, I mean, when you think about the Human Genome Project and how long that took, and you think about now where you can sequence a new genome in 2 weeks. I mean, we've really, the advances are incredible. I did hand sequencing when I worked in the lab. I don't think anybody, probably nobody in my group, none of them are young enough, have ever done that. So, you know, it's great to be an antique. It's like talking, they look at you like you come from Jules Verne's time. So as a first step, we decided to consider insertional effects. So that's kind of where we started. So we thought about this and thought, well, we're spending a lot of time looking at unintended effects. You know, we worry about all these kinds of things. And are we benefiting from our experience? We really didn't think so. Are we really seeing all the things that we thought? We haven't. So we really thought, well, you know, we need systematically to embark in a way of approaching the whole concept of unintended effects. So we considered insertional effects first. And that, because this relates back to the molecular characterization, and everybody does a molecular characterization. And, you know, Paul, and we can argue about how critical it is, but everybody does one. It's a common element for the safety assessment of food, feed, and environmental It's not the entire assessment, but it informs all aspects of it afterwards. And we can talk about it. It'll inform routes of exposure, levels of expression, presence or absence of new proteins. So it underpins a lot of things you can do in the risk assessment. And upstream you can do the molecular characterization, and you can avoid some of the questions that run downstream. So when we embarked on the insertional effects project, we wanted to review insertional effects and their potential to result in unintended traits. So when we start talking about this, this is the whole idea. When you put a new gene into a plant, what happens? Disrupting all these genes, creating havoc, new toxins, horrifying things that you could never imagine, chimeric proteins. So in keeping with the comparative approach, we want to compare insertional effects to other types of genetic changes that are occurring in plants already. So the kinds of things that happen in plants, either through conventional breeding or plants. Because plants have a lot of, there's a lot of plasticity inside those plants anyway. They do lots of stuff. And when you start looking at them and with the genomics tools that we have, we find out that really there's a lot of stuff going on. So has our understanding evolved sufficiently to reevaluate the criteria required for pre-market assessment of GE plants, foods, and feeds? And that was kind of where we started out. So we're going to look at this. We're going to look at this in context and see what's going to come out the other end. And I think we started off hopefully without preconceived ideas. And the idea was to really embark on a structured analysis of what these things meant in real terms. So I was going to invite Jamie to come up here and explain this. She didn't give me the legend for the figures on the side of the slide here. She gave me the slide without the legend. So I'm sitting here going, what the hell is this? But leave it to say that this is an example of the things that can happen during an insertional effects. You can have obviously deletions, and often that happens. You'll get, things will delete out when you put the gene in. You'll get rearrangements around the borders when you get DNA repair mechanisms happening. You can get other kinds of recombination events happen. So generally, when you fire a gene into a plant, things happen, and things will happen around the outside of that gene, and as a consequence of DNA repair and a lot of other things. So they can include insertion of intended DNA, the insertion of additional DNA. You can get other stuff going in. You can get deletions. You get rearrangements. And the insertional effects can result in the expression of unintended traits. So when we thought about insertional effects, we decided to put a tight box around the outside. So many of you, when you're thinking about insertional effects, you're thinking about positional effects or the pleiotropies. So when you've got a single gene, the pleiotropy single gene conferring multiple traits, or position effects. But we really just wanted to talk about insertional mutagenesis, not for any other reason other than to say we had to start somewhere. And it just became far more complex to talk about some of these other things, and it was a lot more difficult discussion, especially position effects and some of the things that really complicate this. So insertional effects are an unavoidable consequence of genetic engineering, but the introduction of unintended traits is not. So just because you put a gene in, just because you have an insertional effect, doesn't mean something's happening. So genetic changes similar to insertion effects occur during movement of transposable elements. Remember Barbara McClintock and her PhD, or her Nobel Prize? So you'll have movement of transposable elements. A lot of plants have them. The repair of double-stranded breaks by non-homologous end joining. The transfer of organelle DNA to the nucleus. These are all things that occur naturally in plants. Plant genomes are constantly changing, and insertional effects will make a relatively small contribution to the final genetic makeup of a given plant variety. When you think of the scope of the genome, when you think about what plants do, when you think about the fact that you have to be a plastic genome because you've got that whole G by E interaction, the genes genetics times the genome times the environment, when you think about all those kinds of things, that plants have to be adaptable. I mean, through evolution, you have to be adaptable to all the insults that are going to happen to you anyway. The conclusions from our project, we're hoping, are going to be used to reevaluate the criteria required for pre-market assessment of genetically engineered plants and the foods and feeds derived from genetically engineered plants. And this is going to be especially useful for retransformance. So and I know we've gone through internationally when we've discussed this, these are difficult words when you come up with your own words. We think about retransformance simply as plants which have been transformed with the identical construct as a previously authorized plant of the same species, which confers the same trait. So it's a pretty specific definition as well. And I know retransformance means different things to different people. You're talking about, GMO being transformed with yet another gene. But this is really what we meant in our context. So this is really about looking at those unintended effects in the context of our retransformance, i.e., plants that we've already, say a potato plant, which you put in a gene which confers insect tolerance, and then you transform that potato plant with the Solanum tuberosum, you transform it with the same construct again. We consider that, we describe that as being our retransformant, so the gene construct is the same. And we've had some discussions about how flexible we're going to be, but I think we're going to start really there. We have some potatoes and we have some apples, which I'm sure many people have heard about, and we're going to, these are things that we're going to have to struggle with. So as I explained to my colleagues, because I know Catherine here is in the room as well, who went through the nature mark potatoes, when we have, you've got potatoes and you've got transformants and you've got a bunch of traits. I mean, between the apples and the potatoes, this could be, for some of the guys in my group, this could be the rest of their careers doing risk assessments on these things. Or we can think about how to deal with retransformants. And it can be, these things can be really resource intensive, obviously, not to mention frustrating and boring. So work's continuing to determine how best to apply the conclusions of this work and to finalize the paper documenting these findings. So we've embarked on the process. Jamie's done an excellent job reviewing the literature, looking at the kinds of things that happen in conventional plant breeding. We've looked at the kinds of possibilities of unintended effects, and we've tried to contextualize all those things together and to think about, well, what does that really mean in real terms? And how do those things fit together? Can we really try and improve the comparative risk assessment? Can we really put at least that aspect of unintended effects into context? So in conclusion, the risk assessment, that's always a great sound, isn't it? And I've got 4 minutes. This is great. I got 4 minutes left. The risk assessment of GE plants is a fairly new science. And we started with the highest level of precaution. So when we started out, people really didn't know. They really didn't know. We embarked on this whole process, and I think if I recall back to some of the people that I met in my early days in regulation, their concept was, one of the ideas behind the novelty concept was that we'd be finished with this. 10 years out, we'd be done, and these things wouldn't be regulated anymore. Novelty was just a way of, they wouldn't be considered to be novel. And I talked to Jim White at the USDA and Simon Barber, who was one of the original authors here, and I think that was really the concept everybody started with. They really didn't think this was going to be a huge deal, and I think maybe people miscalculated a lot of the public interest and maybe some of the discomfort with new technologies that people have. Although if you see all the kids wandering around with cell phones plastered on the ear, wandering across the street, texting while they're coming into oncoming traffic, maybe it's only certain technologies that are uncomfortable with. So with nearly 20 years of experience, can we reevaluate some of our early concerns and put them in perspective? So can we really go back and think about where we came from and we worried about all these things? Can we put these things into perspective now that we've got 20 years worth of experience? What have we really seen? Have we really seen the kinds of things we thought we were going to see? Unintended effects are an outcome of all forms of plant breeding. Do we have the correct perspective for those arising from genetic engineering? So I'm sure if I look around the room, who regulates conventional plant breeding? So do we really have the right perspective on these kinds of things? Canada's embarking on a process to examine some of the consequences of unintended effects and put them in the context of conventional breeding. So Annie's right. We do regulate some aspects of conventional breeding. If you whack something with X-rays or mutagens, chemical mutagens, that's considered to be conventional breeding in the rest of the world. But we really do. Do we have the same perspective on all of those kinds of things? And over the next 2 days, we're going to be examining unintended effects from a variety of perspectives and try and shed light on the concerns and how best to approach them. So I think I'm looking forward to some of the interactions here to hear other people's perspectives, because we've been operating in a little bit of a bubble here, which is why I'm glad Addie brought this up at the OECD meeting. We've been operating in a little bit of a bubble as we've had our discussions inside. It'll be good to enlarge the discussions and hear other perspectives and hear what others are thinking about the route that Canada is starting to embark on now. So sometimes unintended effects are going to happen, and I think Merv and Sam there will be happy to tell you that sometimes things like that just do take place, and you really can't do anything about it, I'm afraid. So anyway, thanks very much, and I got 1 minute and 15 seconds left.